So I want to thank Susanna for inviting me here at the university to tell you the story that I've been working on for over 8 years that I've been in Tom Sudoff's laboratory. And then I will address and tell you what I've been doing in our own laboratory based on the evidence and the results that I've gained over my postdoctoral career. So I also want to take this time to thank the professors that I've spoken to over these past 2 days, because really I had the pleasure of talking to them about their work, and it really sort of highlights how many neurobiological questions that are out there that still remains unanswered, and the approaches today compared to 20 years ago is really quite amazing. So I want to thank you all. So to begin my talk, I wanted to talk to you today about the X11 proteins. I've been studying them for quite a long time, and the interest came from Tom Sudoff's laboratory, in which these are multi-domain proteins. Multi-domain proteins meaning that they can bind to several synaptic proteins, and they can also bind to two genes that are associated with Alzheimer's disease, such as amyloid precursor protein, APP, and presenilin-1, which are the two candidate genes have been associated with familial Alzheimer's disease. So I like to give the talk into two parts. The first part is the role of the X11 proteins in synaptic function, and then the second part will be the role of these X11 proteins in association with Alzheimer's disease. So the laboratory of Tom Sudoff's main interest is really synaptic transmission, how one neuron communicates with another at a specialized site at the synapse, and where the nerve impulse actually activates neurotransmitters to be released from the synaptic vesicle, and they cross the synaptic cleft, in which they bind to, the neurotransmitter bind to the receptor, post-receptor sites of the receiving neuron. So here on the right, it's an electron micrograph of this asymmetric synapse, where you have the presynaptic buton that contains synaptic vesicles where they are docked at the active zone, where they are release neurotransmitters upon calcium trigger, and then they bind to the postsynaptic receptor sites in the postsynaptic neuron. And a large focus of the laboratory is really trying to understand the mechanism behind the synaptic vesicle cycling event, and how really essential proteins that are expressed in the presynaptic compartment actually regulates the several cycles of the synaptic vesicle events, such as synaptic vesicle translocates to the active zone, where they dock at the active zone and are primed for fast calcium trigger release of neurotransmitters, and then the synaptic vesicles gets anisotosed and then returns to this active sort of cycle at the presynaptic end. So along with this sort of very well-governed process is along our essential synaptic vesicle proteins, a lot of protein-protein interactions, which I'm sure you've heard about through Susanna, and I'll give you examples. Synaptotagmin is a very important calcium sensor for calcium trigger release of neurotransmitters. The snare proteins are highly important for the fusion process, synaptobrevin, SNAP25, and syntaxin. But what I will talk to you about today is really on an adaptor protein and their function at the synapse. And it's really interesting because the MINT proteins, the MINT1 protein was originally found was originally identified in association with two other proteins, Cas protein and Willis protein. And this was a tripartite complex that is evolutionary conserved throughout C. elegans. And the hypothesis was that Cas interaction with a cell surface protein, neurexin and neuroligin on the postsynaptic membrane, that this sort of complex is hypothesized to be important for the linkage between the presynaptic and the postsynaptic site. But it's not to say also that this complex is also found at the postsynaptic site as well. And it has several suggestions that it also has some sort of postsynaptic properties. So MINT proteins was originally identified in Tom's lab where his interest was mainly on MUNC18, because MUNC18, he showed that it was an essential molecule for synaptic transmission. So the approach was, what are the binding or interacting partners that can interact with MUNC18 that can actually regulate this presynaptic events? And what we pulled out from this, the yeast 2 hybrid screen are the MINT proteins, in which he named as MUNC18 interacting proteins. And as I previously alluded to before, that the MINT adaptor proteins also binds to two Alzheimer's-related proteins. They bind to amyloid precursor protein at the PDB domain and presenilin 1 at the PDZ domains. And then the question was, what is the role of the MINT proteins in association with synaptic function and as well as Alzheimer's disease? So a little bit about the MINT X11 proteins. I told you that they're named as MUNC18 interacting proteins, but they were actually originally identified as X11, as a candidate gene for Friedreich's ataxia, but that was later ruled out. So in the literature, you'll see MINT1, MINT2, MINT3. There are 3 distinct isoforms, and they're also named X11 alpha, beta, and gamma. In addition, the official name for it right now is APP binding protein A1, 2, and 3. So there are 3 distinct genes. They have a very divergent N-terminus, meaning that only MINT1 and MINT2, which are neuron-specific, can bind to MUNC18. Only MINT1 can bind to the Cas, forming this tripartite complex. This tripartite complex is made up of MINT1, Cas, and Villase. And then they have a very conserved C-terminus that consists of a phosphotyrosine domain in all 3 of the genes and 2 PDZ domains. And as I mentioned, all the PDB domains of all the MINT proteins bind to amyloid precursor protein, APP, and there's been several reports that suggest that the PDZ domains actually interact with several proteins, such as presenilin, neurexin, which is a cell adhesion molecule, calcium channels, and kinesin protein, which has been postulated to have a postsynaptic role. So where are MINT proteins expressed and in the brain? And so here, illustrated in panel A, we did a Western blot looking at protein expression across all levels of the body. So you can see that MINT1 and MINT2 are specifically expressed in the central nervous system, in the brain and spinal cord, whereas MINT3 is really ubiquitously expressed in all tissue. And we use VCP as a control. So on the right panel, what you can see is that we did immunostaining with the MINT1 antibody as well as the MINT2 antibody. And what this shows is that it showed us that these 2 neuronal isoforms are specifically expressed in different neuronal subtypes, but they are not exclusively within neurons, because you can also find MINT1, MINT2 co-localized in the same neuron as well. But for example, that I'll give is that on MINT1, at the level of the CA3 region of the hippocampus, MINT1 also is found staining in the GABAergic interneurons, whereas MINT2 is really highly localized to the CA3 region of the hippocampus. Another example is the level of the cerebellum. MINT1 is highly expressed in the inhibitory Purkinje cell layer, as well as several cells in the granular cell layer, whereas MINT2 is really devoid in this Purkinje cell layer. And then we have the olfactory bulb, the glomeruli in the olfactory bulb, which is mainly GABAergic, but then it also stains the excitatory mitral cells, whereas there's really very small staining of MINT2 in the olfactory bulb. And they're really differentially expressed in all layers of the cortex. So that's also very interesting for us, because you have the same family members, but they're expressed differentially in different parts of the brains. So our goal was really to try to determine what the in vivo function of this family of adaptive proteins. And so our approach was to generate and analyze mice that are deficient in all the MINT X11 proteins by homologous recombination. So the MINT1 knockout was originally made first. We did it as a conditional knockout because based on work from the C. elegans, if you knock out the MINT1 protein, they have dramatic phenotype. There's a significant decrease in survival in the C. elegans. So we decided to sort of pursue the knockout strategy as a conditional knockout. And so what that means is that we took the first exon and we flanked the exon with LOX-P sites with the condition that if you cross them with Cre recombinase transgenic mice, you can exclusively delete this protein by LOX-P recombination. And then we use neomycin and diphtheria toxin as our positive and negative selection. And the neomycin was flanked by FRT sites so that we can remove the neomycin gene if we need to, because oftentimes neomycin sort of interferes with protein expression. So what do we find with the homozygous FLOX gene? The homozygous FLOX gene contains the exon, the LOX-P site, the neomycin gene. And what we found was that the protein levels of the homozygous FLOX was very similar to what we found in wild type. So that sort of suggests to us that our homozygous FLOX alleles did not did not interfere with protein expression. So we went ahead and crossed these homozygous FLOX mice and crossed them to transgenic mice that carry Cre recombinase in the male germline to remove the first exon, and we get complete deletion of the protein of MINT1. And then we can further have a cleaner knockout such that the neomycin is also deleted as well. And so when we first generated the MINT1 knockout, there was really no dramatic phenotype, meaning that the mice survived, and the really small sort of phenotype that you might see is that they're a little bit significantly decreased in regard to their size. And so we did a systematic sort of weight analysis throughout 2 months of age, and we did indeed find a small significant change in their size of the MINT1 knockout compared to the glutamate control. So we then did an immunostaining of the wild-type mice compared to the MINT1 knockout mice at different levels of the brain. We looked at the hippocampus, and you see, as expected, MINT1 deletion is deleted in the brain. We see a higher power, so you see that these are MINT1 staining with GABAergic interneurons, and that this is really deleted as expected, as well as in the olfactory bulb and the cerebellum. And we also looked at MINT2 staining, and MINT2 staining did not seem to change in the MINT1 knockout. And structurally, we did also Nissl staining, and there doesn't seem to be any sort of obvious structural phenotype in the brain. So the question was, we wanted to characterize whether the MINT1 protein actually have a synaptic function. And to address this, we did a collaboration with Dr. Malenka's laboratory at Stanford University, in which they are known to do the hippocampal slice electrophysiology, in which they looked at more specifically in the Schaefer collateral pathway. So the hippocampus is really a unidirectional sort of pathway network, such that the majority of the inputs to the hippocampus comes from the entorhinal cortex, and it synapses to the dentate gyrus, and the dentate gyrus then feeds onto the CA3 region, the mossy fiber pathway, and then the CA3 neurons then synapse onto the CA1 area, and then this is the Schaefer collateral pathway. So what they did was they did field recordings as well as whole cell recordings and looking at the synaptic responses of the CA1 region. So we first looked at excitatory synaptic transmission, looking at 4 paradigms of short and long-term synaptic plasticity, such as pair-pulse facilitation, post-tentanic potentiation. And if there were any sort of changes in release probability, we would see changes in these 2 paradigms, and we didn't see any changes in those two paradigms. If we were to see changes in vesicle recycling, we would see changes in use-dependent depression, and we didn't see any changes in this paradigm as well. And if there were any sort of alterations in postsynaptic receptor function, we would see changes in long-term potentiation. And again, we didn't see any sort of changes across these four paradigms in the excitatory synaptic transmission. But as I mentioned to you before, it seems that MINT1 is a little bit higher expressed in inhibitory neurons. So our next question was, are there changes in the inhibitory synaptic transmission? And here, what they looked at was the pair-pulse depression, and what they indeed found that there was a dramatic change in pair-pulse depression, that there was MINT1, deletion of MINT1 actually regulates the release of GABAergic neurotransmitters. So to summarize from the MINT1 knockout, because they were produced first, was that the MINT1 and 2 are similarly expressed in most neurons, except for the fact that MINT1 is a little bit higher expressed in inhibitory interneurons, whereas MINT2 is highly expressed in excitatory neurons. Deletion of MINT1 does not impair the survival or the overall architecture of the brain. There were no changes in short and long-term synaptic plasticity associated with the MINT1 knockout mice, but however, we saw that in inhibitory synapses, MINT1 actually impairs the GABA release if you compare it to control. So the MINT2, MINT3 knockouts came afterwards, and this was my time with Susanna, that we spent a lot of time in the ES culture room, and we were successful in producing the MINT2 as well as the MINT3 knockout. So different from the MINT1 knockout, take a step back. So similar to the MINT1 knockout, that MINT2 and MINT3 were made as conditional knockout as well. The exons were flanked by LOX-P sites, neomycin, and diphtherotoxin was used as a positive and negative selection. What's different about the MINT2 and MINT3 was the fact that it seems that neomycin was actually interfering with protein expression. And what you can see is that when you see the homozygous flocks from MINT2 or from MINT3, you get deletion of the protein expression. But however, when we cross these mice to transgenic mice that have the flip recombinase to remove the neomycin, we get return of the protein expression back. And then we created a more cleaner knockout such that both the neomycin and the exons are deleted. So our next question was, what are the functions if you remove all three of the proteins from the brain? So from a Mendelian ratio of genotypes from several of the MINT crosses suggests the following. So if you only take the single knockouts and you cross a heterozygous to each other, what you would get is a 1 to 2 to 1 ratio, meaning 25% would have wild type for MINT1, 50% for the heterozygous, and 25% for a homozygous, which means that they survive normally. There's no major lethality. So in the single knockouts, MINT1, MINT2, MINT3, all the single knockouts, they survive fine. There was no obvious sort of phenotypes wrong with them. So we actually went ahead and said, okay, let's just address the double and triple knockouts and not be concerned with the single knockouts of MINT2 and MINT3. So when we did the crossing, what we found was the following. If we sort of did any combination with the MINT3 and then it was on a MINT1 knockout background crossing with MINT3 heterozygous, we also get a 1 to 2 to 1 ratio as expected, so suggesting that most of the mice are surviving at a normal Mendelian ratio. However, it's only when you delete the two neuronal isoforms, MINT1 and 2, that you see dramatic decrease in the MINT1 and 2 knockouts. So we saw 80% of the mice do not survive, and that only 20% survive through adulthood. And so the question was, we did this Mendelian ratio of genotypes at 4 weeks of age. Do they die embryonically, or do they die immediately during the postnatal life prior to 4 weeks of age? And so we did a series of genotypes immediately after birth, and the double knockouts are born immediately after birth, but within 12 hours they seem to die off. And the most obvious sort of phenotype is that there was no milk in their stomach, and then possibly they die of dehydration. So we first look anatomically at very early on newborn MINT knockouts. So most of the analysis now is based on a MINT1 knockout background because due to several reasons, there was a very small percentage of MINT1 and 2 double knockout survival. So in order to increase that sort of percentage and looking at the MINT1 and 2 double knockout, this was going to be on a MINT1 knockout background. And we had done a lot of analysis on the MINT1 knockout background. So what you see here is an H&E of the sagittal section of the head. And so one hypothesis was because MINT has been associated with CAS, especially MINT1, and CAS has a very dramatic phenotype such that they also die early on in postnatal life. One of the reasons why they die is because of a cleft palate abnormality. So we went ahead and see whether there's abnormalities associated with cleft palate, and we found that the palate was normal. And then we did Nissl staining, and our brain architecture was normal. We didn't find any sort of obvious changes. And here we did a MINT2 staining of the CA3 region, and as expected, the MINT1 and 2 double knockout, the MINT2 staining is depleted. And then this is APP staining as a control, so it did not change. And then we also did a histological examination of the adult MINT1 and 2 double knockouts, so 20% that sort of survives. And we didn't find any sort of dramatic anatomical phenotype associated with the surviving MINT1 and 2 double knockouts. So here is a Nissl staining of a horizontal section of the brain on a single MINT1 knockout background, MINT2 wild type, and the double knockout of MINT1 and 2. As expected, MINT2 staining is depleted in the brain. We did staining with APP and RAP3A, which is another synaptic protein, and there doesn't seem to be some changes. So the 20% of the mice that survive, they're not normal, meaning they have dramatic they're significantly smaller. We did a systematic weight curve for the males compared to the females, and both males and females, they're completely, significantly much smaller compared to their litter mates, which is the MINT1 knockouts. And so even though the MINT1 knockout had a little small weight differences, the addition of the MINT2 deletion increased this sort of weight measurements dramatically. So next we sort of looked at the synaptic function, in which we used the hippocampal slice electrophysiology, and what Wade had measured was first the input-output measurements. And so this was an indication of looking at synaptic strength, and so what we immediately found was that the MINT1 and 2 double knockout, marked in red, was significantly decreased compared to the single MINT1 knockout. And what this sort of suggests is that the synaptic strength is decreased, but possibly through a mechanism, a presynaptic mechanism, possibly through the release probability of neurotransmitters to be released at the presynaptic site. So what he next did was he did a 14 hertz stimulation, and what he found from this is that the MINT1 and 2 double knockout had a dramatic, a really significant enhancement in facilitation, and so this sort of suggests that, this result sort of suggests that there's alteration in a decrease in presynaptic release. So they continue to look at this further, and they did parapulse facilitation in the excitatory synapse as well as an inhibitory synapse, and they found that there was an increase in parapulse facilitation. So an increase in parapulse facilitation is meaning there's a decrease in presynaptic release because they're inversely proportional. And we did not find changes in the inhibitory pair pulse depression. They went ahead and looked at spontaneous release in excitatory as well as in inhibitory synapses. They did not find changes in the amplitude in both these synapses. However, there was a significant change in the frequency in excitatory synapse, such that MINT1 and 2 double knockout there was a significant decrease in spontaneous events in excitatory neurons, and we did not find changes in the inhibitory cells, which further suggests that it's most likely a presynaptic event that is occurring in the MINT1 and 2 double knockout mice. So in the 20% that survived that we gave them for analysis, a lot of criticisms could be that the 20% that survived, there could be some compensatory events that might be happening that's what we sort of, the results that we get. So one way to sort of address this was to make a mouse line in which it was homozygous for homozygous flocks for all 3 genes, MINT1, MINT2, MINT3, and that this mouse line would then be crossed to each other to make it a homozygous triple flox line, and that we can take pups of P0, culture them, cortical neurons, and then infect them with the lentivirus that express either a Cre recombinase that's linked with GFP or a mutant Cre recombinase linked with GFP at 3 days in vitro, and then those infected neurons would be the ones that are deleted with they're deleted for MINT1, 2, and 3 if they were exposed to lentivirus that contains the Cre recombinase. And that this sort of model, we can compare the control, which was infected with mutant Cre, and we can compare it to the MINT triple knockout neurons. And so the advantage is that you can delete all 3 proteins at once, allowing for a direct comparison of neurons with the same genetic background. So when we characterized these cultures, what we found was that we were really able to delete the MINT proteins very well, all 6 alleles in the tissue culture, in neuronal cultures. So if you compare the control, which neurons were infected with a mutant Cre, they express MINT1, MINT3, and MINT2. And then in the MINT triple knockout neurons, we really have complete deletion of these 3 proteins, and GDI was used as a control. We also examined the neuronal cultures by electron microscopy, and there doesn't seem to be any sort of structural differences between the control and the MINT triple knockout neurons. They have really normal synaptic structure of the presynaptic bouton and the postsynaptic receptor. Postsynaptic site. So a graduate student actually in Tom's lab did electrophysiology on these MINT triple knockout neurons, and what he found was with the whole cell recording was that there was a significant decrease in miniature APSCs, the currents, in the MINT triple knockout neurons, and there was no changes in the amplitude, similar to what we found in the sliced electrophysiology. And that we also see a decrease in frequency in inhibitory cells and no changes in amplitude. So when there's no changes in amplitude, it sort of suggests that there might be no alterations in postsynaptic receptors Yeah, I'm sorry. These are MINT triple knockout neurons. So these are the ones that were excised because they were excised by infection of lentivirus. Control meaning all 3 proteins are present. And so the next question was, if they're altering presynaptic machinery, is it because they're regulating the readily releasable pool of the neurotransmitters in the synaptic vesicles? And to sort of address this question, he applied sucrose, evoked and measured the synaptic responses in excitatory and inhibitory neurons, and they found that we get a dramatic decrease in sucrose evoked APSCs, but we did not see changes in sucrose iPSCs. So next, what we did was we took these culture neurons, a control meaning they express all 3 proteins, and then a Min triple knockout in these cultures, and there was a significant decrease in all 3 protein expression. And then across the board, we sort of looked at potential proteins that might be altered in these Min triple knockout neurons, proteins such as the proteins that they interact with, such as APP or Cas in MUNC18. And we found that APP did not change, Cas didn't change, Vili's didn't change. However, we did see a significant increase with MUNC18, and it was very nice because it sort of directly correlates that there is an interaction between the MUNC18 and the Min proteins. We also found changes with an adaptor protein, Fe65, and a GluR1, which is a synaptic, postsynaptic receptor. And then along with the changes with Fe65, we saw changes with the liporeceptor protein, LRP. So from these analysis of the MINT knockouts, what we can conclude is the following, is that if we delete the MINT proteins, we get upregulation of MUNC18, and that this sort of alters the presynaptic dysfunction that we observe in the knockouts. Because if we overexpress MUNC18 in culture, we also see a very similar phenotype, such as there's a decrease in EPSC's frequency, but not in amplitude. And at the same time, we observed an increase in Fe65, and this was also very interesting to us because Fe65 is another family of adaptor proteins. They also contain the PDB domains, but they contain 2 PDB domains. And Fe65 also binds to amyloid precursor protein. So to see an effect of Fe65 proteins may sort of suggest that they have a role in association with APP, and that if you knock out the MINT proteins, possibly Fe65 can compensate for the function association with APP. So if we remove MINT proteins, we see an upregulation of Fe65 because Fe65 binds to APP, and that it's been shown that Fe65 can regulate the expression of LRP, and that there was a decrease in LRP expression. So the summary of the MINT knockouts are the following that we found. The MINTs 1 and 2 are functionally redundant proteins and are important for mouse survival. MINTs 1 and 2 are important for presynaptic release of neurotransmitters, possibly through their binding with MUNC18. Fe65 proteins that we saw that are increased may compensate for the lack of MINT proteins, possibly by binding to their common APP binding. And so really the synaptic part of the story sort of ended there, but hopefully I can continue this in my laboratory, and I'll tell you a little bit about what we're going to do in the future years in my laboratory. So the second part that I want to introduce is the role of MINT proteins in association with Alzheimer's disease. And so Alzheimer's disease, as most of you know, is a neurodegenerative disorder. Right now, there's about 35 million people that are afflicted with Alzheimer's disease in the world. It's characterized by progressive memory loss and other cognitive functions. Its most common cause of dementia in the elderly, and the 2 most vulnerable is age and the genetics. And then there are really a dramatic brain changes that is happening throughout the years of an Alzheimer's patient. So here on the left is a healthy coronal section of a healthy brain, and on the right is an advanced Alzheimer's patient's brain. And what you can see is there's a dramatic shrinkage of the brain. You get enlarged ventricles, and you really get the hippocampus is really completely obliterated in the advanced Alzheimer's patients. Along with these brain changes comes two neuropathological hallmarks. One is the neurofibrillary tangles within the neuron, which is an accumulation of abnormal phosphorylation of a microtubule-associated protein called tau. And then a second hallmark is our amyloid plaques. And amyloid plaques are deposits of 40 to 42 amino acids, and they're derived from the precursor protein, amyloid precursor protein. So APP is a transmembrane, type 1 transmembrane domain. It has a large extracellular portion, and it has a smaller cytoplasmic region. And A-beta cleaves from sequential secretases such that you have the non-amyloidogenic pathway in which you have alpha secretase, and alpha secretase cleaves right in the middle of A-beta. It generates a soluble APP alpha form and a cell membrane-associated C-terminal fragment called CTF alpha, also known as C83. And if you get the beta secretase pathway, it's the amyloidogenic pathway, meaning that beta secretase cleaves at the end of the A-beta and then produces the soluble APP beta and then the C-terminal fragment beta, which is also C99 and C89 fragments. Sequentially, after either alpha or beta secretase cleavage, what you get is the gamma secretase cleavage. And what happens is that gamma secretase cleaves at the edge of the A-beta. The non-amyloidogenic pathway produces the P3 fragment, and then in which the gamma secretase cleave after beta secretase produces the A-beta peptides. And it's been suggested that there's another cleavage that cleaves, that releases the APP intracellular domain, and this has been sort of hypothesized to go to the nucleus. So the MINT proteins, as I mentioned before, they have the PDB domain, and all three MINT proteins have the PDB domain, and they bind specifically to the MPXY motif, the very conserved motif in the C-terminus end of APP. And there's been literature from in vitro studies that suggest that the PDZ domain binds to presenilin 1, which is part of the gamma secretase complex. And also in the literature, there's been a vast amount of literature that looked at the MINT proteins in association with APP processing. And there's been really a discrepancy in what they've seen. So, for example, if you overexpress MINT X11, they reduce A-beta secretion. However, if you silence by RNAi, delete the MINT proteins, they also lower A-beta levels. So what is really going on? So we sort of took advantage of our knockout system and crossed it to the APP transgenic mice that overproduces A-beta peptides. So that was our goal. So the goal was, do the MINT proteins have a function in relation to APP processing? And that the strategy was to take these knockout mice and cross them to the APP transgenic mice. But because we had such dramatic phenotype, survival phenotype in association with MINT 1 and 2 or MINT 1, 2, and 3 double and triple knockout, we actually just took the single knockouts, which survives fine, the single knockouts, and crossed them to the APP transgenic mice. So first we looked at the MINT1, and the MINT1 knockouts were crossed to transgenic mice that express the Swedish and Indiana mutations. And what we see is that here we have the MINT1 wild type carrying the transgene, so you see A-beta immunostaining in the hippocampus, and then this is the MINT1 knockout carrying the transgene, and when we quantitate the A-beta pixels in the hippocampal area, we get a significant decrease in A-beta deposition. We also looked at A-beta 40 and 42 peptides by ELISA, and we see that in the soluble as well as the insoluble, that the MINT1 knockout can decrease A-beta 40 and 42 production as well. So then, because of the Swedish Indiana mutation, we had to wait a very long time for amyloid deposits to occur close to 12 months of age. We actually analyzed another APP mouse line. And this APP mouse line is a Swedish, and it also contains the presenilin-1 delta E9, exon 9 deletion. And what this transgenic mice, this double transgenic mice, is that they can produce A beta production as early as 6 months of age in the cortex, hippocampus, and then very little in the cerebellum. So as you can see in the cortex as well as the hippocampus, there's an increased correlation in A beta in relation to age. So what happens when we cross the single knockouts to these transgenic mice? We looked at 3 different ages, 6, 9, and 12 months. We looked in the hippocampus as well as the cortex. We sort of quantitated A beta pixels from the immunostaining. So this is also 6, 9, 12 months. And what happens in the 6 months is that we get a very significant decrease in A beta production if you knock out mints, single knockouts of mints. And this continues to be significant at 9 months, in which MINT1 and MINT2 significantly can decrease A beta production. But now MINT3, we don't see any significance with MINT3. And then in the hippocampus, MINT2 continuously becomes significant to delay the production of A beta deposition. And we sort of see similarly in the cortex, but however, we don't see the significance with the MINT2. And in fact, by the time 12 months comes on, you see a significant increase. So by the time sort of the beta amyloid sort of takes over, it doesn't seem that there's any effect of the MINT proteins. And so in fact, from the conclusion of this data is that the MINT proteins, if you knock out MINT proteins, you only could delay the processes of A beta deposition. We also looked at this by ELISA, A beta 40 and 42, looking at the soluble fraction as well as the insoluble fraction. The insoluble fraction has been known to be what causes the A beta to aggregate, and then we have the soluble fraction as well. And then what we found was that we see very similar correlation to what we found when we immunostain with A beta. And so the question was, why do we see more of a significance with MINT2 compared to MINT1? And the one possible reason is because when we immunostain with MINT2, MINT2 is really highly expressed in the excitatory pyramidal neurons in the CA3 region of the hippocampus, and as well as APP. APP is also very highly expressed in the CA3 region of the hippocampus, whereas MINT1 is really devoid of that staining. You only see very few staining in excitatory neurons, and mostly in GABAergic interneurons. So this might be most likely a role because they are expressed in similar neurons that you have more of a dramatic phenotype with the MINT2. So the next question was, what is the mechanism behind this sort of delay and decrease in A beta production? So we know, for example, that from the amyloidogenic pathway, we see a decrease in A beta formation. So this could be most likely be at the level of beta secretase cleavage, or it could be at the level of gamma secretase cleavage. And to address that, we turned back into this Cree lentiviral system, in which we took the triple homozygous flocks, but now they carry a transgene, this APP Swedish presenilin mutation. We culture neurons, we infect them with the Cree recombinase or the mutant Cree, and then now we can compare the control and the MINT triple knockout neurons that are now carrying this transgene. And we can then look at biochemically at what's going on. So here, again, is a Western blot, the MINT triple knockout neurons that are deleted of MINT1, 2, and 3 compared to the control neurons, which contains all 3 proteins. And this is the protein levels quantitated. And when we looked at A beta 40 and 42 levels, we see a significant decrease in A beta 40 and 42 in the MINT triple knockout neurons compared to control. So we can recapitulate this in culture, and now we can address biochemically what might be going on. Is it at the level of beta secretase or at the level of gamma secretase? So one experiment that we can do to address this is that we can treat these neurons with DAPT, which is a gamma secretase inhibitor. So if you inhibit the gamma secretase, what you would get, you won't get the cleavage to A beta. What you'll get is an accumulation of the C-terminal fragment, CTF beta. And so what we see in the control is that when we blot for APP and its C-terminal fragments in control, you would expect that there's an accumulation of C89, C99 fragments. And this is from COS cells that express C99, so you know that it's migrating at this area. And so when we treat the neurons, the triple knockout neurons, with gamma secretase inhibitor, what we found was that there was a significant decrease in the production of C-terminal fragments. And so this sort of suggests that most likely the changes that we're seeing with decrease in the A beta is most likely coming from beta secretase cleavage. And so we did this a little bit more extensively by applying the gamma secretase inhibitor over time, in which the black line represents the control mice. So you see, as expected, an increase in C99 C-terminal fragments. And when you quantitate the C-terminal fragments in the mint triple flocks, you see a significant decrease, sort of suggesting that there's alteration in beta secretase cleavage. And we also looked at, well, were there changes in base levels? And so this is, we looked at whether base levels were altered, and there was really a small significant change in base levels as well. So the summary of the role of the MINT proteins in APP processing is that if you delete the single MINT1, MINT2, and MINT3, it reduces A beta production and beta amyloid plaque formation. And that the effect is age dependent. It actually only delays the plaque deposition, and MINT2 deletion was the most potent suppressor of A beta production. And the deletion of MINTs most likely is altering the beta secretase cleavage of APP and unlikely the gamma secretase pathway. So in association with the Alzheimer's disease story, so if you sort of delete the MINT proteins, functionally at the synaptic level, we see increase in MUNC18, and the changes in MUNC18 might be responsible for what we see at the presynaptic level. And if we delete the MINT proteins, what happened with APP? We get a decrease in A beta formation, and that most likely it's due to beta cleavage, a decrease in beta cleavage. So the future projects of the laboratory now really focuses on the molecular mechanism underlying the APP MINT biology in Alzheimer's disease, more of a neurodegenerative pathway, and really look at the cellular level what's happening, the connection between the MINT proteins and APP. We're going to investigate the role in APP trafficking and how that alters the processing. We're also going to be looking at biophysically looking at the MINT interaction with APP, because from what we see from preliminary data, it seems like the N-terminus domain and the C-terminus domain actually regulates the binding of APPs, such that the N-terminus has more of a promoting factor that enhances APP binding, whereas the C-terminus can inhibit APP binding. So there is a very dynamic sort of binding properties that are occurring between these two proteins. And that we want to identify potential regulators of MINT proteins. We just started have some initial data that the MINT proteins can be phosphorylated, and whether the phosphorylation of MINT proteins have an in vivo function, such as presynaptic function or as well as APP processing, that would be very interesting to address. So most of this work that I sort of told you today was largely done at UT Southwestern Medical Center under Thomas Sudoff. Dennis was the graduate student at the time who did the electrophysiology. Shunrun and Katsuhiko Tsubuchi sort of established the lentiviral system. And at Stanford University, we've had a long collaboration with them in which Wade Morishita from Rob Malenka's lab did all the slice physiology for the mid-knockouts. And right now, Jia Shen is really a very nice collaboration for the direction that I'm sort of pursuing. And then here's my group at Boston University. I've been there for 1 year, and I'm very lucky to have 2 very senior associates working with me, 2 graduate students and 3 undergraduate students. So thank you very much.